Hello guys, in this video we are going to study about blood sample testing, a topic from FMT, and a lot of questions are asked from it. Right, first of all, what is blood sample testing? In blood sample testing, we know that whether a fluid I have found on the crime spot is blood or something else. We will study the same for semen sample also. It is especially important in case of rape. But in blood sample, in case of murder, homicide, and other types of various things, blood sample is important. Now, to test if the sample is blood, we can do either a color test or a luminescent test, and there is confirmatory test. So first of all, coming to color test, then color test, the main principle is that blood contains hemoglobin and hemoglobin contains heme, which contains peroxidase activity and which react with its agent to give color, right? So there are three types of color test: benzidine test or Adler's test, phenolphthalein or Castle test, and third is orthotoluidine test. Benzidine or Adler test gives positive for blood when if the color is blue. For orthotoluidine test also, the positive test gives a blue color, while for phenolphthalein or Castle test, it is pink is positive, right? And then coming to the luminescent test means we spray luminol or we give UV light to the stains. For example, wash stains. For example, there is a cloth and in that cloth there is a stain, red colored stain, and we want to know it is really blood or something other colored dye that has been put on the cloth. For that, we can use UV light or luminol spray. If that gives a luminescent color in darkness, that means that it is a sample of blood, right? So first of all, let me tell you that these things I told you, luminescent and color test, are not important. I just only gave this and included this in my video to for a sense of completion, right? These things will never be asked in your examination till now, whatever I have told. Only the things I have written in red is actually important and will be asked in the examination. Coming to the confirmatory test, there are three types of confirmatory test. First is microcrystal test, second is absorption spectroscopy, and third is microscopy. Now coming to microcrystal test. In microcrystal test, there are two types of test mainly. First is Teekmanin test. One is Teekmanin test. Teekmanin test. And the second one is Takayama's test. Right. Takayama test. In Teekmanin test, the important thing is, first of all, stain is heme. Heme is the part that is stained in it, plus Teekmanin reagent. If we give Teekmanin reagent with it, then this particular reaction will form brown rhombic crystals, right? This particular reaction will form brown rhombic crystals. Brown rhombic crystals. Brown rhombic crystals will be formed, right? And these crystals are hemine crystals. These crystals are hemine crystals, right? So important thing over here is Teekmanin gives brown rhombic crystals. In question you will be asked that a test was done and brown rhombic crystal was seen, right? And they will ask you which of the following test was actually done. So they will give you option like Teekmanin test, Takayama test, luminol test, and then they will another give you, for example, fluorine test, right? So first of all, you will be able to eliminate last two options, and you will be able to come into options Teekmanin and Takayama test. Then you will get confused whether it was Teekmanin or Takayama. So you have to remember that Teekmanin is brown rhombic crystal. And what about Takayama? In Takayama, the same reaction is done, but if we mix Takayama reagent, we will get pink crystals, right? We will get pink feathery crystals. Pink feathery crystals. So if there is written that pink feathery crystal is present, then it is Takayama test. And if it is written that brown rhombic crystal is present, then it is Teekmanin test. Very important, you have to remember it, right? So first of all, how to remember this? In my times, I use this technique that you can see over here the word man, M-A-N. Teekmanin contains the word man, right? And with man, you can associate the color brown, right? That these things are, manly color. So I made this particular trick. And what about the second one? The second one contains the word ma, right? Ma means female. And if that is female, then you will be associating the color pink with it, right? So this is the way I used to remember it, because in examination you will confuse these things that Teekmanin was brown or Takayama was brown. So you have to remember Teekmanin means man means brown, and Takayama means pink, right? So this is the way you can use this technique to remember. Now coming to absorption spectroscopy. In absorption spectroscopy, nothing much is important, but you have to remember that this is the most reliable test, and it can be used for recent as well as old blood stains also, as well as old blood stains also, right? Now microscopy part is very easy. First, take the blood stain and see in a microscope. If you see this type of RBC, then it is RBC of human, right? And if you see somewhat like this and nucleated, the shape is altered and nucleated, this means it is the RBC of an animal, right? So this is how you differentiate RBC of human and animal. So let me give you a summary of it. It is a very easy part, right? Summary of whatever you have taught and whatever is important. It is very important, right? Only these things are required. First of all, teak mannin, and second important part is Takayama. Teak mannin and Takayama. Teak mannin gives man means brown rhombic crystals, right? They will give you brown rhombic crystal. They will ask you this, brown rhombic crystals. And what about the next one? Takayama ma means pink crystals. Pink feathery crystal. You have to remember these words also. Pink feathery, brown rhombic crystals, right? You have to remember these also. And last is absorption spectroscopy. Absorption spectroscopy is most reliable. So this is everything actually you need to remember. This video, I could have completed this video in these three things only: teak mannin, Takayama, and absorption spectroscopy. Only these three things are important. For the sense of completion, I have taught you everything over here, but nothing else is important other than what I've written in the summary part. Thank you.